Hello, good day. Third year, we are going to be talking about antigens in this video lecture. So for the past video lectures, we were able to discuss the basic principles of immunity, the basic principles in differentiating adaptive versus innate, origins, and development of your T cells and B cells. So in this video lecture, we will be discussing about the natures of antigens, and this is important because we need to understand how the push through or properly in the serologic principles in the future. So you can remember that an immune response is triggered by immunogens. So immunogens are considered macromolecules. So sabi natin, talking about antigens. So, ano yung ibig sabihin pala ng immunogens? So, immunogens in its essence are antigens that are capable of inducing an immune. So, the antigens in its technicality is just a part of a certain foreign material, certain substance that act as a part or structural part of a whole um, organism or whole component or molecule. So we specifically term immunogen as immunogen for the antigen or the antigen that is capable of inducing the immune response. So there can be antigens that can bind to antibodies but are not enough to trigger the immune response. So just a quick trivia. So our antigen in its essence can be referred to as a generator of an antibody. So your immune response, your immunogens specifically react with corresponding antibodies or sensitized T lymphocytes. So your T lymphocytes, remember in our discussion, it can evoke an immune response initially and allow to stimulate the B cells to produce the antibodies required specific antigen. So the two terms immunogens and can be frequently used interchangeably. But in this lecture we will refer immunogen as so your antigens will combine with specific receptors on the surface of antigen sensitive cells so that it will stimulate the whole process of immune response. So anong ginagawa? Yung cells na nakaka sense or nakaka-recognize ng ating antigen will differentiate into lymphocytes, which can mediate the cellular immune response, or which can also happen to be formed into the antibody producing plasma cell. So, yun lang. Foreign substances can either be immunogenic or antigenic. And we also have what we call as antigenic determinants or what we refer to as epitopes. So these antigenic determinants, always remember that these are structures that are part of the whole antigen that actually react with the antibody. So yun yung part ng antigen that reacts with the antibody. Kasi hindi naman ang whole antigen ang magbabind. So yung specific part of the antigen that binds with antibody is, or a T lymphocyte receptor is the epitope itself. So tandaan lang yung concept na hindi lahat ng surface parts of your antigen are antigenic determinants. Only yung prominent determinants lang or surface of a protein part of the antigen can be recognized by our immune system. So malalaman natin na meron lang specific types of molecules that can trigger our immune response. So ano yung mga different definitions natin of antigens? Meron tayong tinatawag na immunogenicity, antigenicity, allerogenicity, tolerogenicity. So yung sabi natin, immunogenicity is the ability of the antigen to induce humoral or cell-mediated immune response. So in this situation, we will call the antigen as an immunogen. So ano ba yung major properties of our immunogen? So dapat yung immunogenicity as a whole, the ability of the antigen which can stimulate the body to evoke a specific immune response. Tapos immunoreactivity naman, it is the ability of our antigen to combine with a corresponding antibody or sensitized lymphocyte. So these are the different specificity definitions. So ano bang ibig sabihin natin ng specificity? So specificity is a cardinal feature of the adaptive immune system. Di ba sabi natin, ang advantage of adaptive immune system is that na-recognize na ng immune system natin siya before. That's why yung response niya to that specific na a foreign substance is very coordinated. So yung specificity will refer to the immune responses that are directed toward that specific antigen. And that specificity is because of the lymphocyte antigen receptors that can bind to one molecule of that antigen or foreign substance but will not bind to another molecule even with minor structural difference lang only. So yun yung very specific part of the immunogenicity or recognition of antigens. So example, gaano ba ka-specific yung antigen? So for example, this is the part of the antibody that will bind to the epitope ng antigen. So kung ito na antigen, for example, si A antigen can only bind with antibody 1 kasi yung structure specialized for that antigen is only capable of binding that specific antigen. So kung ganito na yung ating antigen B2, hindi na siya pwedeng mag-bind kay antibody 1 kasi yung structure niya will not be recognized by the antibody 1. So ganun din, antibody 2 yung magre-recognize lang kay B2 antigen and kung si si C antigen ganito, only that antibody that is having the structural recognition molecule for that epitope can bind that specific antigen. So yung specificity exists both in immunogenicity and immunoreactivity. So yung specificity is the basis of the immunologic diagnosis and immunologic therapy as well as basic feature of adaptive immunity. So ito, the immunogen is the antigen that induces specific immune response. So ganito ang itsura ng recognition. Up to, depende kung memory or kung antibody production. So for example dito, meron tayong antigen, tapos na-recognize siya ng ating antigen presenting cell. Tapos, after nagkaroon ng recognition, yung antigen presenting cell will trigger certain na mga cells to multiply or proliferate. So during this time, ito na yung tinatawag natin na activation phase. Tapos during the activation phase, this stimulator cell will trigger the lymphocytes to become differentiated plasma cells that are antibody producing. So the antibody that will be produced in the plasma cell phase will be specific for the antigen that was recognized sa recognition phase. And then after that, magre-release siya ng antibody that will now bind to the specific antigen. And once the antibody specific for that antigen will form an immune complex with that antigen, nagkakaroon tayo ng release or triggering of the inflammatory process through either humoral immunity or cell-mediated immunity. And there will be elimination of antigen. So yun na yung effector phase. And what happens is that there will be death of the organism or that foreign substance that bears that type of antigen na na-recognize natin since the recognition phase. So lahat ba nung cells that are being triggered by our antigen presentation will be plasma cells. So hindi, yung iba sa kanila nagiging memory cells which will retain the memory of that antigenic structure ready for another future exposure. So, ganun yung specificity ng ating immune response. So again, the antigenicity itself is the ability to combine with antibodies or cell surface receptors. Tapos, lahat ng molecules that possess immunogenicity will possess the antigenicity but not vice versa. Haptens are parts or types of antigenic structures that are antigens but cannot invoke or induce an immune response. So anong ibig sabihin nito? All immunogens are antigens but not all antigens are immunogens. Kasi hindi lahat ng antigens kayang mag-stimulate ng immune response. And yung tinatawag naman natin na sabi natin kanina, meron tayong antigenicity, immunogenicity, allergogenicity. So yung allergogenicity is the ability of an antigen to induce allergic response. Hindi lahat ng antigen can be from a foreign material that is a pathogen. Some of them are just allergens. For example, pollens or dust mites. So they activate a specific type of humoral or cell-mediated response as allergic manifestation. So, kung may na-smell ka na pollen, it will trigger an allergic response. So, may na-detect tayo na antigen from that allergen, but the type of immune response that happens will be an allergic immune response. So, anong example? Yung allergens natin, when recognized, will or can induce anaphylaxis. So, ano ibig sabihin ng anaphylaxis? This is an example of a severe immediate hypersensitivity reaction because of rapid generalized mast cell granulation. So, minsan, kung meron tayong immunogenicity triggering an immune response, so ano nangyayari? Gusto nilang patayin yung foreign substance that contains that specific antigen. Pero if ang na-trigger nito na antigen is an allergic immune response, meron tayong concomitant na generalized mast cell degranulation. And when that happens, there will be generalized vasodilation, constriction of bronchial smooth muscles, increased production of mucus. So that is not generally directly related to apoptosis of the foreign substance, but formulation of a generalized na allergic response. So yung antibody deposition that is bound to the antigen in the extracellular matrix. For example, ang antigen or allergens, flower powder or yung pollen, seafoods, ganon. So instead of having basta apoptosis lang, magkakaroon tayo ng mast cell degranulation. Tapos some immune complexes will trigger inflammatory cells and yung tissues ng host ang nagkakaroon ng injury. Kasi generally ano naman, hindi naman delikado yung allergen. Nagiging delikado lang siya kasi nagkakaroon tayo ng hypersensitivity reaction towards that allergen. So ganun ang isa sa characteristic definition of an antigen being an allergen. And next is tolerogenicity. So this is the concept of the capacity to induce a specific immunologic non-responsiveness, whether in the formation of antibody or in the cell-mediated branch. So these tolerogens, they induce immunologic tolerance. So pag sinabi natin immunologic tolerance, so ma-remember niyo sa T-cell development, it is the unresponsiveness to an antigen that is induced by prior exposure to that antigen. So pwedeng nagkaroon tayo ng exposure to that antigen. Tapos, ang nangyari, because of our previous exposure, nagkaroon ng desensitization ng ating immune system. So, hindi na niya tinitrigger or nag-i-invoke ng immune response against that specific na antigen. So, what happens is that pwedeng that antigen is introduced in very small amounts eventually hanggang sa na-recognize na siya as non-foreign. So when we are being introduced with that specific substance that contains a telogen, hindi na tayo nagkakaroon ng immune response against that antigen. So yun yung tinatawag natin na immunologic tolerance. Okay? So what are the different chemical it if an antigen is made up of protein and has higher molecular weight, this they can be coupled with a carrier. So, ano nangyayari if meron silang partner na carrier? Pag ganun, nagiging immunogenic na sila. So, anong example natin? We can have what we call as some drugs or chemicals that are partnered with a certain carrier and are now immunogenic. So ano pa ba yung other factors that are effective in the function of antigen? So meron tayong tinatawag na foreignness, foreign. Kung foreign masyado ang antigen, kung mabilis ba siyang ma-degrade or degradability, molecular weight of that antigen, the stability of that antigen, and the complexity as a whole. So, bakit nagmamatter ito ng mga characteristics of that antigen because it determines if that antigen is an immunogen or if its immunogenicity is high. So remember last time na our complement pathways can sometimes be triggered by exposure to a certain structure or part of a structure ng foreign substance. So the degree of foreignness of of an antigen can determine its property of being recognized as non-self. So, paano yun? Kung malayo talaga yung structure niya from most of the antigens in our body, so there is a certain recognition na this is a foreign substance kasi walang ganitong klase ng antigenic property ang ating mga tissues. So, the immunogenicity somehow depends on the foreignness of a molecule to a greater extent. So, yun yung isa sa mga reason bakit mabilis magkaroon ng donor rejection ang transplant tissue na malayo ang HLA characteristic compared with the recipient. So, what are the requirements of immunogenicity? So, it depends on the conditions of the system. So, the antigen, the mode of immunization or immune response, and the sensitivity to detect that immune response. So, ito yung sinasabi natin kanina, foreignness, the degree of discrimination of ourself and against non-self. So, yung foreign molecules should be immunogenic. So example, if there is an albumin isolated from a rabbit serum, tapos in-inject siya sa same rabbit or another type of rabbit. So since they are still rabbit, it will not generate antibody formation unless there are problems with HLA differences. Pero kung yung albumin na yun, in-inject mo to a different form of mammal or higher vertebrates, there is a large chance or higher chance that there is an antibody production that will happen depending on the dose of the antigen na injected. So aside from foreignness, yung ating molecular size or molecular weight, so the immunogenicity will require a specific minimum size. So hindi porket foreign and protein or complex protein, it is already automatically immunogenic. So, paano kung mga 5 Daltons lang? So, hindi pa siya enough to be detected by our immune system. So, the ideal immunogen weighs approximately 100,000 Daltons. So, a very high molecule, example, hemocyanin, it has a molecular weight of 6.75 million. So, it is highly immunogenic. And if low molecular weight molecule naman, it is non-antigenic or feeble. So they render antigenicity or immunogenicity by absorbing some inert particles like bentonite or kaolin. So ang remember natin na large foreign molecules are better immunogens. However, yung sinabi natin kanina, meron tayong tinatawag na haptens. So these are low molecular weight antigens. But when they bind to a carrier molecule, they behave as immunogens. And if that hapten is chemically linked to a larger molecule, which is a new surface structure, the hapten itself will now function as the antigenic determinant. So first, meron na tayong foreignness, molecular size or molecular weight. Next naman yung tinatawag natin na chemical complexity or complexity. So the degree of a chemical complexity of a certain antigen, if mas complex, mas immunogenic. So the immunogenicity increases with structural complexity. So yung complex proteins, they are better antigens than large repeating polymers like lipids, nucleic acids. So they are poorer immunogens. And the complexity of a certain antigen is somehow parallel to the genetic constitution of that animal. Respond to that type of antigen will vary the genetic makeup of the recipient. So, anong example? Pag sinabi nating polysaccharide, medyo good immunogen lang siya, pero hindi siya very good immunogen compared with proteins. Pero for lower forms of mammals or lower forms of animals, that pure polysaccharide will, that pure polysaccharide injected to a certain organism, not necessarily of higher or lower form, but to the degree of different contribution of that animal to that substance, doon nagkakaroon ng difference in response. So yung polysaccharides daw will be a good immunogen sa human beings, a very good immunogen sa mice, pero not so much when injected to a guinea pig. Ganon. So, yun ang ibig sabihin ng complexity din. So, paano ba ang possible methods of antigenic administration? So, the type of antigenic response or the ability of an antigen to induce an immune response can depend on the dose and the mode of administration. So, we will talk about some characteristics of your antigen. So, ano yung sabi natin kanina? So, dapat foreign to the immune system. So, meron tayong tinatawag na Burnett's clone selection theory. So, the non-self that has never had contact with lymphocytes during embryo period has higher degree of. So, kung hindi siya nakasama, for example, yung mga pathogens, hindi naman tayo nagkaroon ng contact ng ganun with our lymphocytes, so mas mataas yung degree of foreignness niya with our immune system. So, ano ba yung mga different substances? Pwedeng heterogeneous substances, allogenic substances, autoantigenic components, and these are the different kinds of substances that can invoke degrees of foreignness to the immune system. So, pag heterogeneous yung hindi talaga natin same species form of antigens. Allogenic substances, so mga antigens that can be found on some humans but are not from our close HLA relationship individuals. And autoantigenic components naman, so these are antigens that can invoke immune response. So, anong example natin? Denatured IgG in patients suffering from rheumatoid arthritis can become an antigen to induce production of antibodies. So, doon nagkakaroon ng development of rheumatoid factor. So, you will appreciate more the rheumatoid factor, even though I think you have discussed this partially in your clinical microscopy or AUBF. You will appreciate this more during discussions of autoimmune disorders. So this is an example of a denatured IgG. So still an autoantigenic component, meaning from our body. And when this is denatured, madedetect pa rin siya ng ating immune system and we will produce an antibody against. So antigens can or minsan nilalagay natin sa mga metabolic products or weakened some part of foreign organisms that we can harvest and allow our body stimulate an immune response against that antigen. So um, another characteristic of immunogens are the degradability. So pag mas mabilis ma-phagocytose ang isang antigen, mas immunogenic siya kasi mas mabilis siyang ma-process ng ating system. So bakit? If an antigen can be recognized um, sa ating system, so there are some antigens that are sufficient enough to stimulate an immune response. So when they are being destroyed by our effector cells, some part of that antigens can be processed and then presented to other T cells. So when that happens, binigay na mismo sa T cell, so that will initiate an immune response. So ang T cell na yung magpo-process na information and will invoke either further B cell differentiation or getting ready for immune response. So we have what we call antigenic determinants or epitope. So yun yung minention natin kanina, that they are the portion of the antigen molecules that are specifically recognized by the antibody or the antigenic receptor of lymphocytes. So a protein antigen can have about 5 to 15 amino acid residues, and a polysaccharide antigen may have 5 to 7 polysaccharide residues. So yung mga epitopes, yun yung key portion of immunogen that is the determinant site. So they are the molecular shapes or configurations that are being recognized by the antibodies. I mean by the B or T cells or the antibodies. So yung ating epitopes can exist repeating copies, or some epitopes can also have differentiating specificities. So, ganito ang itsura. So, ganyan. If we will do a 3D complex protein na visualization. So, again, your epitopes, they are the sites on within antigen which the antibodies react. So, meron silang certain molecular shapes or configurations that are being recognized by the B or T cell. So, meron tayong different types or structures of antigenic determinants. Meron tayong sequential or linear, and meron din conformational. So, yung ating single antigen, specific antigen, can have different epitopes. And the antibody is directed to epitopes. So, ganun ang role epitopes of that antigen. So, pag nagkaroon ng change in the antigenic determinant, so kahit pa same lang ang antigen, it can influence the specificity of that antigen. So, for example, ito. There can be arrangement and conformation which will affect the specificity of an antigen. So, if may amino acid na nag-change, once na makikita nyo yung itsura ng amino acid exposure. So, pag na-change ang arrangement and conformation of that specific antigen, nagkakaroon din ng changes in specificity of that antigen. Meron din tayong tinatawag na antigenic valence. So, the natural antigens mostly are with polyvalence antigen characteristic. So that is a total number of determinants which can be bound by the antibody or an antigenic receptor. So yun yung ibig sabihin ng antigenic valence. So please take note by that portion of the antibody, what that we call as the um, that area of the antibody that binds that specific epitope is yung hypervariable region. So it forms the complementarity determining region. So yun ang part ng antibody that is concentrated with the parts that bind to the epitope. So meron tayong tinatawag na structures, yung sabi natin sequential or linear determinants and conformational determinants. So yan yung mga types of antigenic determinant structure. Pag sinabi nating sequential or linear, so yun yung sequence of amino acid. Pag sinabi nating conformational, so non-sequential polypeptides or polysaccharide on the surface of the molecules. So conformational determinants. So these are formed by amino acid residues that are not in a sequence but are spatially juxtaposed in the folded protein. So ganito itsura. Hindi sila sequential, hindi sila linear, but they are adjacent in such a way that then they can be bound by that specific part of the antibody. So this can happen when there is folding of one chain or multiple chains that will bring exposure of the amino acid from different segments. Makikita nyo naman of this linear sequence. And it will bring them in close proximity to each other so that they are easily recognized together by an antibody even though they are not in a linear sequence. Next, pag sinabing sequential or linear determinant, so medyo straightforward, yun talaga yung group of amino acid residues that are recognized as the epitope. So it is a single chain or a sequential amino acid fragment. So ganito ang itsura. So conformational determinant can be it cannot be detected by the antibody or the immune system if there is a loss of proximity. That can happen during denaturation. Yung linear determinant naman, kahit pa nagkaroon ng denaturation of the protein, since iisang sequence lang yun, pwede pa rin siyang detect ng ating antibody with better exposure kasi hindi na siya inaccessible. Next, so neo-antigenic determinants. So these are the types of determinants that are created by proteolysis. So if may part of the antigen that was proteolyzed, so nagkaroon ng exposure to a certain part of that antigen, tapos na bindan yun ng antibody, so that happens, that becomes as a new determinant. So next, pwede rin na magdepende on the types of cells that are recognizing antigenic determinants. So ganito ang difference between T-cell epitope and B-cell epitope. So pag T-cell epitope, syempre TCR ang nagre-recognize. Pag B-cell epitope, syempre BCR na receptor nagre-recognize. Mostly, ang nature ng mga T-cell epitopes are short peptides, and your B epitopes naman, B-cell epitopes are proteins, polysaccharides, about 5 to 15 amino acid residues or 5 to 7 monosaccharide. And yung size naman ng residues ng T-cell epitopes can reach 8 to 17. Tapos yung T-cell epitope, more of linear, and yung B-cell epitope, more of pwedeng either conformational or linear epitope. So, B cells recognize epitopes that are easily accessible. Pwede rin sequential, pwede rin conformational. Pero yung T cells, they recognize internal linear peptides. So it can be of any position to the antigen. So, meron tayong different common determinants in fact. So, may mga bacteria that will contain epitopes that are common antigens. That's why pag pa-recognize natin, alam natin na bacteria sila. Tapos, there are also what we call na concept ng cross-contamination. So, there are some common antigen determinants that are existing in between different microbes. So, once na may common antigen ang isang organism, which also exists with another organism, pwede natin yun ma-recognize ang antibody natin even though yung antibody present is specific only for one type of antigen. I mean, foreign substance. So, ang tawag doon, cross-reaction. Example, typhoid bacillus, meron tayong anti-typhoid serum, anti-AG1, and anti-AG2. So, si para-typhoid serum, meron siyang antigenic determinant AG2. So, since meron tayong anti-AG2, kahit na yung antibody natin sa serum is just for anti-typhoid, but because it shares, the para-typhoid bacillus shares a common determinant with typhoid bacillus, pwedeng magkaroon ng cross-reactivity. Si anti-AG2 with para-typhoid bacillus. And yung anti-AG2 na na-produce for para-typhoid bacillus, pwede rin mag-react with the AG2 of typhoid bacillus, vice versa. Ngayon, it is important because cross-reaction may lead to wrong diagnosis. So, pwedeng because of cross-reaction, pagkaroon ng So now we will proceed with the different classifications of antigen. So we can base it on immunogenicity. So meron tayong tinatawag na complete antigen and incomplete antigen. So yun yung HAP10, low molecular weight antigens. So pag HAP10 can only possess immunoreactivity when it is paired with a carrier, it can obtain the immunogenicity. So, ganito ang itsura ng HAP10 with a carrier. So, di ba, antigenic determinant na lang talaga siya once paired with a carrier. So, what are the different sources of antigen? Meron tayong sino-antigen, allo-antigen, auto-antigen, and heterophile antigens. So, yung sino-antigen, so they are the types of antigens that are found in more than one species, and they are capable of inducing an immune response. So, pag sinabing sino, it means foreign or other. So, na example, pathogens like bacteria, viruses, fungi, parasite, mga toxins, toxoids, and these are toxins that lost its toxicity but can still invoke an immune response. So example, tetanus toxoid and diphtheria toxoid. So pag xenoantigen, totally different. Ganito example, bacteria, fungi, hepatitis, o viruses, ganun. Next, yung heterophile antigens. So other name, Forceman antigen. So these are common antigens that are shared by different species between human and animal or microbes between different species of microbes. So example, M protein ng Streptococcus. So it bears a common antigen determinant with the basement membrane of kidney. So what is a danger? The Streptococcus can be recognized by our immune system as foreign. So it will develop an immunoglobulin. So pag nag-bind siya nag-create siya ng immunoglobulin with the M protein, there is a chance, since it bears a common antigen determinant with the basement membrane of the kidney, na yung mga immunoglobulin na na-form supposedly for streptococcus lang alone is nagkakaroon ng binding with basement membrane of the kidney. So with that, nagkakaroon ng pathologic process ang ating tissue. Next, yung tinatawag natin na alloantigen or isoantigen. So it is present in one individual for that specific species but not in the other. So example, antigens of the red blood cells, yung ABO system and RH system. So it is governed by the HLA system or the human leukocyte antigen. So this relates or is significant to transplantation and is important in immune regulation when we do transplantations. So example, bumalik. ABO system. So antigens A, B, A, B. Ganun. So anong antigens O? H antigen. Next, autoantigen. So this is when there's a release of a sequestered antigen. So example, lens protein, once it is released to the blood, it can induce immune response to induce inflammation of the lens. And when there is a change in the molecular structure of autoimmune tissues, for example, denatured IgG, I think na-mention to kanina, if nag-denature siya, it will be degraded and it will be induce a production of antibody.
So that's it for your antigen lecture. I hope you learned a lot and study your books. Thank you.